Okay, so here's our membrane, again, in the yellow, inside, outside. These little sections in the middle, they kind of look like cookie sandwiches to me. These are our channel proteins. Okay, so this is what's going to allow substances to pass through the membrane. That makes sense? So I'm gonna click some of these boxes here real quick. And so one of the things that we want to realize when we're looking at this is we have differences in concentration gradients. Okay, so these substances, they're not in equilibrium chemically. So think back to the beginning of the school year, right? We're talking about transfer processes, active transport, passive transport, right? Passive transport is movement from high concentration to low concentration, right? When you put food coloring in water, what happens? Yeah, it spreads out. You don't have to do anything to do that. It happens on its own, right? And so naturally, these substances are going to want to do that. Your sodium right here is at 145. Nm, little m, big M is millimoles. It's just a measure of concentration, how much stuff we have, okay? So we've got 145 millimoles of sodium outside, but we've only got 10 inside. Naturally, what wants to happen is this sodium out here wants to flow inside because it goes from high concentration to low concentration, right? Now, it can't do that because it can only flow through these sodium-gated channels. We know cell membranes do not let charged particles pass through them. Okay, that's one of the things that makes them selectively permeable. So the only way the ions that can go through are through these channel proteins. And right now, the channel proteins are closed. Oh man, there we go. Okay. So at rest, we have a concentration gradient. And so we have more sodium outside, and we have more potassium inside. So this is just that we're sitting at rest right now. Does that make sense? And this is going to be really important because this is going to allow this process to happen spontaneously without having to put energy into it. Energy is going to come into it later. But right now we have this set up so it can happen spontaneously, right? So first what I'm going to do is I'm going to stimulate the neuron, and we're going to go ahead and see if we can plot out the steps of what's happening with the ions. So here we are. We're going to stimulate the neuron. This would be you get stung by a bug. Somebody pokes you with a stick. Yeah, and they get the little hammer and they hit you in the knee. You go to the eye doctor and they're like shining the light in your eyes and stuff like that, right? You take a hearing test and you're hearing the buzzer or whatever it is going off in your ears, okay? So we are stimulating the neurons. So those would be our stimulus. So here's the wave right here. I'm going to go backwards here just a second. Actually, just reset. There we go. Okay. All right, so we're back here. Here's our proteins. Let's stimulate the neuron. We can see the wave of stimulus, and we're going to talk about that here in just a second. Okay, so the wave came and has reached this part of our membrane. What is the first thing that you notice that's changed? Right, the red ones, the sodium channel proteins, open. Now these channel proteins are voltage gated. So when you have changes in charge, and we're going to look how that works after this, they're going to open, right? You don't have to have a molecule come and bond with the protein. So the sodium channel proteins open. So that's going to be our first step. All right, put my little one right here so we know. Okay, So sodium opens first, and then sodium is going to start coming in. Right, now let me bring my concentration back up. Now, okay, you remember sodium right here was at 145? Now we've dropped, right? We're a little bit lower than that. Sodium right here was 10. Now it went up a teeny tiny bit. So we did have a change, right? Yes. This one? No. This one I can't, unfortunately. Well, I can try this. I can go like that, but then this is just going to sit here. So this is how much sodium we have in there, okay? And up here, we have 144.99955. So it went down a little bit, but it went down enough, okay? Now, what was the next? Here, I'm going to go back here for just a little bit. So the sodium's coming in, all that other stuff. Our concentrations went down. Okay, so what has happened next? Right, so we had these ones open, and now we have these ones open. So we have our potassium channel proteins, so that's step 2. Potassium channel proteins open, letting in potassium. I'm sorry, letting out potassium. And so if I let this go forward a little bit, now the potassium numbers are changing. This one was 140, now it's 139.99, so on and so forth. This one was 4, it went up to 4 point blah, 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 1, so it did go up a little bit. It's gonna go, the numbers aren't changing that much, but they don't need to change that much. We're talking about the like molecular level at this point. So really tiny changes have really big effects, okay? So I'm gonna go forward a little bit more. Keep watching over here. What do we see now? Right, so the potassium channel is closing and the sodium channel closes also, okay? So let's put that one down. 3. So the channel proteins close, that's going to stop the flow of ions going across the membrane, right? And then as I go forward, the numbers are going to start to go back to where they were. Like this number right here, it's at 144. It's going to go up to 145 again. Our numbers in the inside are going to reset themselves also. Here, I'll just do this. See, we're back at 145 here. This one is slowly getting back to 140. Well, now we're back to normal, right? So we are at the state where we were before we stimulated the neuron. Does that make sense? Okay, so review the steps. Number 1, stimulus comes through, causes sodium channel proteins to open, sodium comes in. Immediately after that, the calcium or the potassium channel proteins are going to open, the potassium is going to go out. They're going to let the ions move in whatever direction they need to go, and then eventually they close. Does that make sense so far? Folks at home, y'all doing okay? Okay, so now, okay, does everybody have this stuff on the side here? Yeah. Yes. And Na is sodium. All right, everybody has this? Y'all are good? Because I need to clear it so we can see the next parts. So now, okay, I'm going to make the concentrations go away because we're feeling pretty good about that. I'm going to bring the charges in and bring this chart up. Okay, so charges and the potential chart. So right here where it says membrane potential, you can think of it as the charge. Okay, and so we've got charges on the outside, we've got charges on the inside. It's positive outside of the membrane, it's negative inside the membrane. Okay, this would be known as our resting potential. I'm going to stimulate the neuron here just so we can kind of get it going. Okay. You see this flat line right here? That is the resting potential. Do you see a line? Okay, that's fine. This is our resting potential. Now, our resting potential is somewhere in the neighborhood of let's say somewhere in the neighborhood of negative 72 millivolts. So this is, if your nerves are not being stimulated, this is where they're sitting. Okay. Now, when we stimulate them, let me come back here for just a second. With a positive end and a negative end right here, we say the cell is polarized. Okay, that's just a fancy way of saying we've got opposite charge. We've got charges at opposite ends. Okay, one side is negative, one side is positive. Does that make sense? So right now our cell is polarized. Now we're going to go through this again. Here's the wave. We've got our sodium ions coming in. What do you notice happened with the charges, the pluses and the minuses? Right, they go away. And then they're quickly replaced with the opposite. So they flipped places, right? Now the inside is negative and the outside is positive. Y'all with me so far? So all those sodium ions came in, that changed the concentration. It also changed the distribution of positively charged particles outside versus positively charged particles inside. Something to keep in mind. We have negatives. There are negatively charged particles on both sides, but they never move. The charges are determined by the movement of the positive ions. So we don't ever indicate the negatives. We just have to know that they're there. Does that make sense? Did you have a question?
Yes. Did the charges go through one of the channels, or is the NA? What's NA stand for?
So Na, the sodium.
Or was the sodium positive itself?
The sodium itself is positive because it's an ion. So while the first time we looked at it, we were physically exchanging particles, but those particles have charges. And so as the particles move, they're going to change that charge across the membrane. Okay. So, all right, so we've got this next part of the graph right here. Let me get my different color. Okay, so we've got this part right here. The graph went up, right? You'll see that? Looking at the graph, what happened to our charge? Right, it increased. It became more positive, okay? This process right here, when the sodium is coming in, is known as, my arrow, depolarization. It was positive outside, negative inside. We have changed that. We had made them equal to each other and then slightly positive inside. And so we changed that polarization. So we depolarize the membrane, okay? This was polarization. This is depolarization. Now we'll come back over here. So at this point right here, the potassium proteins have opened. Potassium's leaving the cell. So now We have fewer positively charged particles inside. They're all going back outside. Notice how our charges are reestablished, yes? We go back through. Now, if you notice, it's kind of hard to tell. The pluses and the minuses get a little bit bigger. Yes? Because the total number of charges outside of the cell is greater than the total number of charges, positive charges, inside the cell. Yes. Yeah. It's more of which one has more of the positives. Because remember, the negatives don't move. So those concentrations never change. Since we're changing the positives, we've got more positively charged particles. Like I always think of them as marbles with little plus signs on them. And then floating around, but we don't care, are a bunch of little marbles with negative, like little minus signs on them. And so what I'm doing is I'm shooting marbles through through these little channels, right? These are just like little chutes I'm shooting marbles through. I'm going to count up the number of marbles I've got on both sides. And on the outside, I'm going to have more positive marbles, right? And so the negatives, if you kind of think of it like the positives and the negatives, we've got these positives outside, right? And we've also got the negatives. And we know negative and positive cancels each other out. Well, we're just going to have more positives. So the overall charge outside is going to be positive. And the opposite is going to be happening on the inside. Does that make sense? Okay. All right, so, yes, sir.
This is because of the aromatic system, but I see that the potassium and sodium both get dissolved when they leave, when they enter it. Is that happening, or is that just
It's okay. For this particular scenario, they make them disappear. So we're not cluttering the space with all these dots. Okay. Everything's kind of technically everything is dissolved in water. If we're zooming into this level, at this molecular level, all of those little particles would all still be here. This whole space would just be filled with particles. Okay. They're just they're fading away to keep the simulation clean, so we can keep track of stuff. Yeah. No, yeah, it's just a simulation. Okay, so now the charge has gone back down. It has become negative again. This process is known as, put my next arrow. This is polarization. Now it's kind of hard to see. Down here at the bottom, we've actually gone a little bit lower than our starting line. Okay, now we're in the neighborhood of about negative 75 or so. Going lower than the resting potential is known as hyperpolarization. Right. And so hyperpolarization, that's going to put us in the neighborhood of negative 75 to negative 80 millivolts.
Does that happen every time?
Yes. All right. We good so far? Now, this whole process we've been going through, this little button here has been gray. I can't click it. Nothing's going to happen. Okay. So while this process is taking place, we cannot stimulate the neuron. Now, this number is slowly going back up. I still can't press this button though. Now I can. Okay. And so can I move this? Because I think I put this in the wrong place. No, I can't. Okay. Then it goes away. Okay, so this section right here, okay, where we could not press the button, we could not stimulate the neuron. This is known as the refractory period, which is a fancy way of saying the rest period. Nap time for your nerve cells.
So what happens, like we use the example of hitting the knee, right? What happens when that person just keeps hitting it? If you can't stimulate that neuron.
If you try to stimulate the nerve during the refractory period, nothing will happen. Now, here's the thing. If you look at the bottom, this is time in milliseconds.
Oh, just that.
Yeah, so like you yourself or your doctor would have to have really, really good reflexes to hit, like to tap your knee and then tap it again inside of that refractory period. Okay. I mean, it could happen, yes. And if you stimulate a nerve inside of the refractory period, nothing happens. Okay. And so if you watch like doctor shows and stuff like that, and inevitably somebody's in the hospital and they're in critical condition, and everybody's like, what are we going to do? And they have the like machines and stuff hooked up to them, and then you see the like beep, beep, beep. That they're measuring this electrical activity. And so that little flat part in between the beeps is the refractory period. That's natural, right? Now, you can, in some cases, you can shorten that. If you need to increase your heartbeat or increase neurological activity, you can shorten that period. The problem is you can't maintain that accelerated state indefinitely, because eventually your nerves and your muscles and everything will tire out and then they just stop working. Yeah, well, Jake, did you have a question?
How would you lower the refractory period, like make it shorter?
Well, you know, hold on to that one. We'll look into that a little bit more as far as how the body actually shortens that before I just sort of like start spouting some stuff off. So I'll drop that. We'll look at that here in just a second. Yes, sir.
I thought that you can almost stimulate the cells sometimes. Is that just when like stimulate the node sometimes? Is that just when the refractory period is so short and it doesn't really help anything?
Yeah, it can be, yeah. So you're like tapping into that ability to shorten that temporarily. Yeah, so they were in that accelerated state for a certain period of time, and then their body was just like they just couldn't anymore. Okay, so once we get outside of the refractory period, we're able to stimulate the neuron. And so if I do that, we're back at the resting period. Here comes the wave again. We go through the whole process all over again, right? Can't do anything. We're waiting for the refractory period to end before we can stimulate the neuron. And then we can just keep going. Does that make sense? Now, okay, are we all good with these terms and stuff like that right here? All right. Now, there's some, I want to draw your attention to this little sort of stair step looking thing right here. This is showing us something called the threshold potential. And the threshold potential is the charge at which you have to, it's the charge you have to reach in order to have an action potential take place. And that's somewhere in the neighborhood of negative 55 millivolts. So with action potentials, it's all or nothing. If you hit 55, you go through this entire process. It doesn't matter what the stimulus is. So neurologically speaking, as far as your nerves are concerned, if somebody sticks you with a pin or they lightly touch the hair on your arm, your nerves are going to go through this process in the exact same way. The differences come in with the synapses and how the endocrine system works and can either amplify or dampen those signals. If you get less than negative 55, the nerves don't fire. You don't go through this process. And basically, you don't really even notice. Jake. Very good. Yes. So you're going to depolarize the cell a little bit, but there's a certain point that you have to depolarize past, and that's negative 55. And so if we here, so I'll reset. We'll pull up the chart, stimulate the neuron. Okay, so it pops up right here really fast. If we didn't go past 55, then this would just pop right back down and it would be flat. Nothing would happen. Does that make sense? Okay. So, action potentials. In this zone right here. So this is one section of the membrane, all right? And if you picture in your head a neuron has a cell body with the dendrites at one end, and then it has a long tendril-like structure, that's the axon, and then it has the axon terminals at the end, right? So right now what we're looking at is a cross-section of an axon, okay? Or we can think of it like that. Action potentials happen all across the nerve cell membrane as it's going all the way through. So it starts at the dendrites, goes through the cell body into the axon to the terminals. And then you have the synaptic activity. And then it starts over again. Okay. What ends up happening then, if we started at one place, how does it keep going all through the cells automatically? Actually, let me back up first before I talk about that. So one thing that isn't illustrated on here is another protein. It's an active transport protein called the sodium-potassium pump. The sodium-potassium pump actively moves sodium and potassium against the concentration gradient. Okay, so y'all go around town or around the county or whatever, and you see those big water towers. They look like medicine droppers that are upside down, stuck into the dirt, right? So the purpose of the water towers is to provide water during peak times, when a lot of people are turning their faucets on all at the same time. Those water towers have little sensors. And if demand reaches a certain point, the valves open up, or they go out and they can trigger them. Like NBU has a little control center. They can go through, they can manually trigger it, right? That's going to release the water that's up at the top of the tank. It's going to flow down by gravity. It's going to go into the main pipes so we can wash our dishes and flush our toilets and take our showers and stuff like that. And once demand goes back down, people stop using the water, there's a pump in that tank. That pump turns on and it refills the tank, right? So then the next time we need it, all we have to do is just open the valve. Gravity does the work for us. Does that make sense? That's what's happening in our cells. So when we're resting, we have filled that water tank. When we need it, we just open the valve, that's that channel protein, and we're gonna let gravity do the job. Now, in our case, it's not truly gravity, it's the concentration gradient. So we're setting up a concentration gradient in order to, when we open those gates, the particles move how we want them to on their own. Okay, so your nerves don't need energy to do that initial blip thing, right? It's the refractory period in the middle. These pumps move sodium out and potassium in against the concentration gradient. And so basically what happens is your ATP comes in, you've got some sodium from the inside of the cell that sits in these little spaces right here. So it sits like this. Then your protein goes like this. And the sodium leaves. And we've got potassium right here that goes in on the other side, right here. And then it flips like this. And it ejects the potassium. And then sodium comes back in. And then it goes like this. And basically, it just sort of flip-flops back and forth, forcing sodium out and potassium in. Once it goes back to the concentrations that it's supposed to, those pumps shut off. Once we have filled that water tank, that water pump shuts off, the valve closes until we need it. Does that make sense? And so these are our water pumps that force the sodium and the potassium where we need them, at the levels that we need them. So when we need them later and we open those valves, they move on their own. We don't have to turn on any other pumps or anything like that. Okay. So that's why this particular protein is important. Okay, so I'm over here. Okay, actually I'm not over here. I am over here. Okay, so let me blow this up real quick. Okay, y'all know what this is, right? Right. So it's a very coordinated movement. You're sitting in the stadium, you're watching it come around. When it comes to you, like there's certain cues that you wait for before you stand up, right? You wait until it comes next to you, and then you stand up. You go through the motion. While you're going through the motion, the person next to you is watching you, and then they go through the motion. And then when it moves away from you, like 2 or 3 people down, you sit down, and you're not going to just randomly pop up again, because then it just sort of breaks, right? And so this is basically what is happening in the cells. Okay, so remember, we said that there's a stimulus that starts all this stuff. So somebody has to start the wave in the stadium, right? Well, and that's true. So you hope that you can get enough people around you to get it started, and then it's sort of, so that would be like the threshold potential. You need to get enough people to go with you for it to keep going. If you don't, then you're just standing around looking foolish. And everybody else is, you've got 1 or 2 people that are also looking foolish. Okay, so this is basically what is happening with your cells. So now we're back to this. Okay, so we are sitting right here where it says 70 millivolts. We've got the little circles, that's our sodium. We've got our little triangles, that's our potassium. Here's our channel proteins, here's sodium, here's potassium. Okay, we're sitting right here. But look what's happening to the person next to us. They're starting to stand up. So they're about here. We're watching, we're getting ready, but we're not standing yet. Okay. Now, when I move to here, this person, like they're committed. They're standing up at this point. So now, like we're getting ready. We're not quite standing yet, but we're about to. We're like leaning forward in our chair. Here, let me get some. So this is where we are. And this is the person sitting next to us. Okay. Now, here we are. The person standing next to us is standing up all the way. They're like this, right? Where are we going to be? Right here. Yeah, so like we're here. right? We're not all the way up, but we're definitely out of our chair. What is this person doing? So this is the person that's sitting right here. They're sitting there watching, they're getting ready, right? Maybe they're kind of getting out of their chair, but they're still basically in their seat, right? Y'all with me so far? So now here we are, we're basically all the way up, or like 99% of the way. The person standing next to us is all the way up. They're going to actually, they're starting to come down. Okay, right here. The person over here standing next to us, they're about here-ish, right? We get to this part. Now we're the one standing all the way up, starting to come down. The person sitting next to us right here, this is where we've hyperpolarized. Where are they? They're sitting down. We are on the way down. The person over here next to us, they are all the way up, right? Now, we get to the end, number 6. Here they are, they're done. They're resting. They're sitting back, they're watching it go, right? We are almost all the way down. We're like here-ish. We're settling into our seats. And then the person next to us, they're coming down. The person next to them is standing, and so on and so forth, right? So as these voltages change in one place, they affect the voltage in the next step in the chain, which is going to initiate this process. And so as long as we have this voltage change right here, we're going to have it here, which means we're going to have it here, and it's going to keep moving down the line. Once we get to the synapse, things get a little bit different. We release those neurotransmitters. They're going to bond with proteins at the other neuron and restart this process. Yes, sir.
So like use the analogy of the wave, is there anything that could like cause like one horse not to stand up at all, or like not fully get up?
Yeah. If there's nerve damage. If you're paralyzed from the waist down, for example, that would be like, so if we go back to the wave example, you just remove 2 entire sections of the stadium. And so it goes around and then it just stops. And so then the rest, like the wave doesn't keep going with the rest of the people. They can try, but it kind of like it doesn't work all that well because you're trying to like time it and stuff like that. So that could be one. So depending on what you look at, you may see different numbers, but it's around -55. Yeah. Like here, the lower the hyperpolarization threshold is -80. This one has the highest threshold, the peak threshold that it gets to is +35. But like on our simulation, it was 25. So the numbers, depending on what you read, the numbers can be different. So that's why I was saying like in the neighborhood of. It can. Nothing. So like if you're going, if here's our line, if we don't hit that threshold, it just goes like this. Yeah. So like here's the line up at the top of the screen. It just kind of goes bloop, and then it stays flat. And as far as you're concerned and physiologically recognizing that, nothing happens. You don't notice it. That's a check to make sure that you don't react to literally every single stimulus that you come across. It has to hit -55 in order to continue this process. If you get -53, nothing happens. You do not trigger this process from occurring. And all the rest of the cell stays at resting state.
So is the threshold potential the charge of the cell, or is that like the amount of like millivolts in the like stimulator?
It's the charge of the cell. We change the charge of the cell by stimulating it. If we don't stimulate it enough, then the charge doesn't change enough, so nothing happens. It dies at that segment.
You're like not standing up far enough on the way. You just kind of like go partially up.
Yeah, or like if there was an entire section that were just like, nope. And so it came to them and they're just like, I'm just kidding. And then it's just like, well, crap, now what do we do? And so then it just dies. Okay.
That's exactly what it would be like when I stood up to try and do the wave.
And everybody else is just like,
Went along and it was just us standing and nobody decided to stop.
okay, questions about that? So what we need to be able to do going into the project that we launch on Friday is being able to explain this process. Okay, so if we put this all together, you stimulate a nerve, which changes the resting potential of the cell membrane, opening up sodium ion channels. That's going to allow sodium to come into the cell, depolarizing the cell membrane. If you depolarize it enough to cross the threshold potential, you will initiate an action potential, which will allow all the rest of the sodium to come into the cell. That's going to signal the potassium channel proteins to open, allowing the potassium to leave the cell. That's going to repolarize your cell membrane, hyperpolarize it past that resting potential. That'll bring us into our refractory period where we can't stimulate the cell. Once we get past the refractory period, we're able to stimulate that nerve cell and start the process over again. So you need to be able to explain that particular section with that level of detail. Questions from the audience? Questions from the folks at home? Okay, well, let me